So hello everyone. So let's get started with the session. We have some people online and hopefully some people will be joining in some time. So the first talk in this session is GenStore, an in-storage processing system for genome sequence analysis. And Nika, who is a PhD student at ETH, is going to present. Please go ahead.
Thanks for the introduction. So hi everyone, I'm Nika, and today I will introduce GenStore, a high-performance and storage processing system for genome sequence analysis. Genome sequence analysis is critical for many applications such as personalized medicine, outbreak tracing, and evolutionary studies. Genome sequencing machines extract smaller fragments of the original DNA sequence known as reads. Read mapping is the first key step in genome sequence analysis that aligns reads to potential matching locations within the reference genome, and for each matching location during an alignment step finds the degree of similarity between the read and the reference genome by calculating an alignment score. Calculating alignment score requires computationally expensive approximate string matching to account for differences between the read and the reference genome. Read mapping performs this expensive computation on large genomic datasets containing millions of reads. Therefore, read mapping is both computationally expensive and incurs high data movement overhead from storage. There's been significant effort into improving read mapping performance by devising several heuristics, hardware accelerators, and filters that prune reads that do not require this expensive computation. While these approaches address the computation overhead in read mapping, they do not address data movement overhead from storage, whose impact becomes even larger after addressing the computation overhead. Our key idea is to filter reads that do not require expensive alignment computation inside the storage system to fundamentally reduce data movement overhead. And examples of such reads that wouldn't require expensive computation would be exactly matching reads or reads with no matching location in the reference genome that can skip the alignment. However, filtering reads in SSD can be challenging because of various read mapping behavior across different genomic datasets and the limited hardware resources in the SSD. By addressing these challenges, we introduced GenStore, which is the first in-storage processing system for genome sequence analysis that addresses both computation and data movement overhead. And our analysis shows that it performs significant performance and energy benefits compared to state-of-the-art hardware and software baselines. So that was a high-level overview of my talk. Let's discuss some background on read mapping so we can dive into this work in more detail. Read mapping performs this expensive computation on large datasets, and the search space in the reference genome can be very large. For example, a human reference genome can include more than 3 billion characters. So to find some potential locations to do this computation in the reference genome, read mappers typically use an index structure that includes k-length subsequences of the reference genome called k-mers, and the locations of these k-mers in the reference genome. State-of-the-art read mappers typically exhibit various heuristics to work around this expensive alignment computation and typically consist of 3 main steps. And the first step, which is called seeding, we determine the potential matching locations or seeds in the reference genome. To do that, the mappers extract various k-mers from the read and look up these k-mers inside the index. If a k-mer hits in the index, the mapper marks the locations of such k-mers in the reference genome as potential matching locations or seeds. To further reduce the search space and therefore reduce the cost of computation, a second step called seed filtering or chaining prunes some of these seeds in the reference genome to which the read is predicted not to align, using simpler approximation of alignment score. And finally, the third step, which is alignment, determines the exact differences between the read and the reference genome using the computationally expensive approximate string matching. So that's a brief background on read mapping. In our work, we try to experimentally show the potential benefits of ideal in-storage filtering by performing a case study on real-world genomic datasets using various read mapping systems, each of which exhibiting several optimizations. And we also analyze these on state-of-the-art SSDs with different types of interfaces. And we make several observations. First is that the ideal in-storage filter significantly improves performance by addressing both computation and data movement overhead. And second, filtering outside SSD relatively performs lower performance benefits. So first, it does not address that data movement overhead. Also, it must compete with read mapping for resources throughout the system. We also observed that for various state-of-the-art hardware accelerators that significantly address the computation bottleneck in read mapping, the impact of I/O on performance becomes even larger. So based on these observations, our goal is to design a storage filter for genome sequence analysis in a cost-effective manner. And we have 3 design objectives based on that. So first is to provide high-end storage filtering performance to overlap the filtering inside the SSD with the read mapping in host system, and also to support reads with different properties and different degrees of genetic variation. And we also aim to design a system at a low hardware cost. And to that end, we introduced GenStore. And our key idea is to filter reads that do not require that expensive computation using some simple accelerators in the SSD controller, and send the reads that require more substantial process to host system with, or like some accelerator. However, the 2 challenges that we discussed are different read mapping behavior across various genomic data sets and the limited hardware resources available in the SSD. Let's take a look at different types of genomic data sets, and based on that, the filtering opportunities that we leverage and the read mapping behavior according to them. So currently sequencing machines produce one of 2 types of reads. Short read sequencing technologies produce reads that are highly accurate but short, for example, up to a few hundred DNA characters. And long read sequencing technologies generate reads that are less accurate but long, from 300 to millions of DNA characters. So based on that, we leverage 2 filtering opportunities based on 2 classes of reads that wouldn't require expensive computation. One are first class is exactly matching reads that do not require expensive approximate string matching, which is done during alignment. And examples of this frequently happen in short read sets with low sequencing error rates and reads that have low degree of genetic variation. And the second class of reads that can be filtered simply are reads that do not match to any location in the reference genome, and therefore they skip alignment. And frequent examples of this can happen in long reads that have high sequencing error rates, or long or short reads that have very high degree of genetic variation, for example, some viral samples or bacterial samples. So for that, we introduced 2 simple in-storage filter designs, GenStore EM for exactly matching reads, and GenStore NM for non-matching reads. Let's take a closer look at GenStore EM. So GenStore EM is an efficient storage filter for filtering exactly matching reads inside the SSD using simple operations and without requiring expensive alignment. But that is very challenging to find these reads in the SSD because of large number of random accesses for various k-mers within each read. And that is challenging in NAND flash-based SSDs because of expensive random accesses to the flash chips and the limited DRAM capacity which is in the exists inside the SSD. So to address these challenges, we introduced the concept of read-sized k-mers to reduce the number of accesses per each read. Therefore, instead of extracting several k-mers out of a read, we consider the whole read as one k-mer. And that read-sized k-mer enables us to find exact matches only with one index lookup per read. And the second idea that we leverage is to sort these read-sized k-mers to avoid random accesses to the index structure. And that would allow us to find exactly matching reads by sequentially scanning the read set and the index structure. So I will dive into it in more detail with a simple example of a read data set that has only 10 characters. So usually they can be like few hundred characters, but for simplicity here, I'm just using a read with 10 characters. So we introduced 2 data structures. First is a sorted read table with reads and the ID of each read, and a sorted k-mer index with read length k-mers extracted from the reference genome, and the locations of these large k-mers in the reference genome. And each of these data structures are sorted alphabetically based on each read and k-mer. So we scan through these data structures based on the comparison result of the read and the k-mer. So if the read and the k-mer are identical, we detect the read as an exactly matching read that can be filtered from further more expensive computation. And we go to the next element in both arrays. And if the read is bigger than the k-mer, we conclude that the k-mer doesn't match to any read, then we go to the next k-mer. And finally, if the read is smaller than the k-mer, since so far we've been sequentially scanning, we conclude that the read does not match any location in the reference genome, and therefore it needs to go to the host system for full read mapping. So by these two data structures, we avoid random accesses, and we can do filtering only using simple low-cost logic. So here I'm showing that despite the key benefits that these two data structures provide, their size can be very large. And that is because of the large number of unique k-length, read length k-mers. So these k-mers are typically like around 10 times larger than the k-mers used in the baseline read mappers. So the size of each k-mer is also larger. So we try to reduce the size of the data structure by replacing these k-mers with strong hash values of each read-sized k-mer. And by that, we can reduce the size of this data structure by 3.9 times. So this data structure is still larger than typical index using conventional read mappers, but this solution is feasible for our in-storage processing system due to larger capacity we have available in SSD and the higher internal bandwidth of modern NAND flash-based SSDs. So here's a simple high-level overview of GenStore EM's exact matching design. So we store these two data structures, sorted read table and sorted k-mer index in the NAND flash arrays, and these are evenly distributed between different channels, dies, and planes, and leverage the full internal bandwidth of the SSD. And we have a comparator logic inside the SSD controller. So in the first step, we just sequentially scan these data structures in a batched manner and write them in small batches in the DRAM inside the SSD. And in the second stage, we perform exactly matching filtering using that simple comparator. So these two steps can be run in a pipelined manner, and during the filtering, GenStore EM sends the unfiltered data for further processing to the host system so that can happen concurrently also. So now let's take a look at GenStore NM for non-matching reads. So GenStore NM is an efficient chaining-based filter in storage for filtering non-matching reads. So please recall that chaining or seed filtering was the second key step in read mappers that aims to prune some potential matching locations in the reads and prune the reads that don't have any potential matching locations in the end. However, performing this operation in SSD can be challenging because it requires, for every seed in a read, performing many iterations of dynamic programming algorithm. And that can be very challenging for long reads because they can have up to millions of characters. So in our design, the approach we take is to devise a lightweight chaining filter that would selectively filter the reads that have only a small number of seeds. And for the reads that have a larger number of seeds and require more complex chaining, they would directly be sent to the host system. And that is based on our observation on various real-world genomic data sets on various species. And we observed that here, for example, the alignment probability of a long read in a long read data set as a function of number of seeds per read. And we observed that reads that have a large number of seeds, typically with high probability, would align and would not require in storage filtering because they wouldn't be filtered. So then we conclude that with a simple lightweight chaining filter in SSD, we can filter most of the non-aligning reads in SSD at low hardware cost. So for more details on GenStore NM's design, please refer to our paper. Now, let's take a look at our results. So we analyze various read mapping systems, which are state-of-the-art software and hardware read mappers for short and long reads. And we also analyze the performance impact of GenStore when these state-of-the-art read mappers are integrated with GenStore operating inside the SSD, filtering the non-matching reads, and see how much benefit that provides. And we also analyze various types of SSDs, low-end SSD, medium-end SSD, and a high-end SSD. So here I'm showing the performance benefits of GenStore EM for a human read dataset with short read dataset with 80% exactly matching reads for software and hardware baselines. And we observe up to 2.5 times speed up compared to the software baseline, and up to 3.3 times the speed up compared to the hardware baseline, and an average 3.9 times energy reduction. We also show the performance benefits of GenStore NM on a long read dataset with high sequencing error rates and a very high degree of genetic variation compared to the reference genome that we're testing, in which 99.7% of the reads within a line. And we observe up to 27.9 times the speed up compared to the software baseline, and up to 19.2 times the speed up compared to the hardware baseline, and on average 27.2 times energy reduction. So we analyzed the area and power consumption of GenStore by synthesizing it at 60 nanometer technology node, and observed that for an 8-channel SSD, the area of GenStore is 0.2 millimeter square and 26.6 milliwatt power consumption. And we scaled down the area to lower technology nodes and compare it to a 14 nanometer interprocessor, and observed that GenStore takes only 0.006% of its area, and compared to the 3 ARM cores used in a SSD controller, it takes less than 9.5% of the area. So I quickly skimmed through other results that we provide in the paper before summarizing this talk. So we analyzed the effect of read set features on performance, such as data sizes and various filtering ratios. And we also analyzed the performance benefit of implementation of GenStore outside SSD. And we observed that in some cases, it provides actually performance benefit compared to the baseline due to more efficient streaming accesses, but its performance benefits compared to GenStore inside the SSD is significantly lower. And we also provide more detailed characterization of read mapping workloads for various species. So to conclude, I provide a brief summary of our talk again. So there's been significant effort into improving read mapping performance through various approaches. And while these approaches address the computational overheads, they don't address the data movement overhead from storage. And our goal is to improve genome analysis performance by addressing this overhead. And our key idea is to filter reads that do not require expensive alignment step inside the storage system to fundamentally reduce the data movement overhead by addressing these 2 challenges, which are various read mapping workloads across different genomic data and limited hardware resources available in SSD. And to that end, we propose GenStore that provides significant performance and energy benefits compared to software and hardware baselines. So thank you very much for listening. Yeah, thanks.
Questions?
John Demme, Microsoft. Can you better characterize the amount of computation that GenStore would require? I'm thinking, would a few extra ARM cores in the flash controller be enough? Would a small FPGA be enough?
Yeah, that's a great question. So GenStore requires very simple computation inside the SSD, so that depends how much area overhead or energy overhead you want to incur. So we use accelerators for the lowest area and energy overheads there. But we haven't directly studied that, but we're very interested into analyzing it. And I think that a small FPGA integrated in the SSD controller can perform this computation, because it requires simple comparators, and also for the chaining one, we significantly reduce the size of logic required. So yeah, that's also one of our interests, to try to see if we can implement it in real systems.
Thank you.
Yeah.
Any more questions? Are there any questions on Zoom? So let me ask you a question then. So in this work, your assumption is that you have already, the whole data is available for you. Mm-hmm. So is there a possibility of the data that is changing? Because you're trying to pre-sort the data and store it, right? So what is the cost involved with that?
That's a very great question. Actually, we provide a lot of detail about that in the paper. So for the reference genome, that is something that is not that frequently generated. So obviously the pre-processing for that can be amortized. And when the read data sets are generated, they follow some pipelines. So it's a sequencing, which is from the original DNA sample of a patient or an individual or whatever sample. We do sequencing to generate these ACGT characters. also after the base calling. And then we envision that GenStore's pre-processing can be done as part of this pipeline. And we measure the throughput of that pre-processing compared to the sequencing and base calling throughput. And we observe that it provides significantly higher throughput, so it can fully be overlapped with that in a pipeline without changing the frequency of that pipeline in a sense.
I see. I see. Any other questions?
Hi. So this workload is really for big data and also for very huge batch sizes, right? Because you basically have a lot of reads to align to some genome, right?
Yeah.
And so doesn't that mean you need a lot of memory just to do the processing itself? Because you need to scan the entire genome with respect to any read.
Yeah, great question, actually. So as I was explaining in my talk, that was one of the main challenges that we tried to address. So we need usually we need very large memory space to serve these random accesses. But since all our accesses are sequential, we can, at each point in time, bring very small batches, sequentially scan these batches inside the small DRAM in the SSD. And after sequentially scanning them, throw them, bring the next batch. So we, for one of these use cases, for example, we only need a few hundred megabytes of DRAM inside the SSD. Yeah.
Yeah.
Hi, Ais Ribson from UC Berkeley. Really like this.
Thanks.
I was curious, I think for a lot of the long read data, it's oftentimes used for de novo assembly. Is that something you've thought about also targeting, or are you just focusing on, and then maybe in the future, or just focusing on like comparing to an existing genome?
Oh, that's a great question. Yeah, we are definitely interested into exploring various expansions of this work to, let's say, de novo assembly and other, let's say, metagenomic applications also. So I do not have the exact designs of that in mind, but based on initial thinkings, we think that some of these primitives can be, again, used there because for de novo assembly, you would need to do these string matchings or comparisons. And we hope that we can use this design, at least with simple expansions of it or tweaks to it. Yeah.
Okay, then let's thank Nika again.
Thank you.